Fair.
Yeah. So you do that every day?
I go up the hill.
And so it's like there's no clinical reason.
But I know.
You get to the South Street Bridge, that way you hit the road. They like to know if, I'm like, okay. Oh, nice. Still most of it. You guys have no problem doing it every other.
All when they lose their.
Yeah.
In a big place station.
I tell everybody, that's how I start. We do the whole like. all the way down, and I'm like.
Well, it's like this animation.
Yeah.
Really listen.
And
I probably will.
that's great.
Yeah.
Look at my paper.
Yeah, I know. It's like,
Like, yeah, look at the sheep.
So funny because we.
My mom was about assistant.
And for sure, about horses, so it's one of the few drugs we can use in horses is Bactrim, yeah. And they get tons of stroke effective. They got stuff in my gut fermenter, so they get. So. It's so you have to give. An order, right? Injectable, it's just like. And like.
Right now, they're doing a bunch of their. Redoing it. Dog is like taking.
Was,
That's actually super. That's right.
I mean, this was.
accident.
Hello, everybody.
Right.
All right. Thank you all for coming to the first of the 2025 to 2026 Eastern Pennsylvania ASM branch meeting. I'm very excited to have as our first speaker for this year, Dr. Carol Sobotyk, who's coming to us down the street from the University of Pennsylvania School of Veterinary Medicine. She got started as an undergraduate at the Federal University in Santa Maria in Brazil, where she remained for her master's and PhD. And then she moved to the United States to do a residency in clinical parasitology at the University of Texas, A&M, and has been at Penn Vet for a few years since 2022. And I have not seen a talk on worms in a while, and I have not seen a talk on eyeworms at all. So I'm really excited to hear about the content today. So thank you for being here.
Thank you. Parasites. Yeah. So yeah, I'm Carol Sobotyk. I did my residency at Texas A&M University, not University of Texas, because they are enemies. Just to be clear, one is like a Longhorns and the other one is the Aggies. I run the clinical infectious disease lab with my amazing colleague, Dr. Cole. I'm responsible for the parasitology side of the lab, and he, of course, the microbiology stuff. Okay, let's talk about emerging zoonotic eyeworm infections. And why I decided to talk about this first, because it's an emerging disease, zoonotic disease. Second, because it's my favorite parasite, at least at the moment. And third, because we don't know a lot about it. And so far, we do not have case in humans, at least not here in the US, but probably in the future, we are gonna see some. So to start talking about worms in general, and specifically worms that infect the eyes of humans and mammals, it's important to know there is a variety of different species that can infect the eyes of those hosts. And the reason why those parasites are located in the eyes are different. Could be specific neurotropism, like a Toxoplasma gondii, like this first picture here, or could be just a result of life cycle, larval migration, like Toxocara species, Toxocara canis or Toxocara cati, that in humans, they go directly to the eyes and can cause ocular larva migrants. Or could be the primary location of those worms. In those cases, on the primary location of the parasites is in the eye, what does mean? The adult worm is located in the eyes of mammals or any other host. We call them eye worms. And we have a number of different species that are considered like eye worms, but I am more interested in zoonotic eye worms specifically. And in the US, we have a few species that they are emerging zoonotic disease, including species from the Onchocerca genus, which include Onchocerca lupi and Onchocerca gutturosa. Onchocerca lupi can infect mammals, basically, including humans. It's endemic in New Mexico and all the surrounding states. We have few cases in Texas as well, including in humans. Onchocerca gutturosa, it's an eye worm from production animals, but we have few reports in humans as well in the US. And we have a Thalassia genus. Thalassia genus, we have basically two species in the US. Thalassia californiensis, as the name says, it's prevalent in the southeast of the US. And Thalassia calipida, that it's a very recent worm that was introduced in the US, and it seems to be endemic in the Northeast region. So I'm going to focus on Thalassia calipida specifically because I'm very curious about, and because it's a very, very recent endemic zoonotic eye worm that I believe it's endemic already. But until like few years ago, 2020 and before, was considered like an exotic parasite. So the number of cases in the US right now, it's increasing very, very fast. What all those parasites, all those eye worm species have in common, they are vector-borne nematodes. What this means, they are transmitted by vectors specifically. Each one has a specific life cycle and different vector, but what they have in common, they need the vector. So without the vector, we are not going to have the disease. So all the control measures and prevention measures should be focused also on the vector. And why we are seeing like this increasing numbers of cases and also distribution of this parasite. I believe it's a combination of a very complex factors. We are seeing like Thalassia calipida, for example, was not prevalent here in the US, was not detected at all in the US until few years ago, and now it's everywhere in Pennsylvania, New York, and New Jersey. So, of course, we need more research to talk about this, but there is some hypothesis why we are seeing those high numbers here. And most of the time, the reasons are related with the two different factors. Abiotic factors that is related with geography or increasing temperature and etc., or biotic, like land use practice, changes in human demographics or behavior. So we are sharing the same space with the domestic animals, wild animals, and all those hosts can play a role in the transmission and epidemiology of the disease. So all those factors we always need to consider when we are talking about vector-borne disease in general, but specifically vector-borne nematodes. But what is Thalassia calipida? So Thalassia calipida, it's a very beautiful worm. It's a very cool worm. Was previously named as the Oriental eye worm because it was first detected in Asia long time ago. And after, not long time ago, was like 1960 something, the first case. But since then, a lot of cases show up in Asia, and after that in Europe. The first case was in Italy, and after that a number of different countries in Europe as well, and more recently here in the US. And there is this this parasite is a vector-borne disease, so it's transmitted by a vector. And what is cool about this specific vector, it's that the vector is a fruit fly. So it's a fruit fly that occasionally feeds on lacrimal secretions of mammals. The hypothesis is why this fruit fly eventually feeds on lacrimal secretion. It's because this fly needs a high level of protein, specifically for reproduction. And to have all this amount of protein, they look for lacrimal secretion in mammals. So saying that, it's also very interesting because only the male fly feeds on lacrimal secretion of mammals, not the female one. So it's a little bit different than other vector-borne diseases, especially transmitted by mosquitoes, that most of the time the female mosquito is responsible for the transmission of the disease. Thalassia calypta is the male fruit fly from one specific genus named Portica species. And we have 2 species so far confirmed to be involved in the transmission of Thalassia calypida. Portica variegata here in the US and Europe, and Portica okadai also in Asia. And this parasite has a number of different hosts, so they can infect a number of different mammals, including carnivores and lagomorphs. But domestic dogs seems to be the main host for this parasite. All the information that I'm saying here, it's based on research from Europe. So we do not understand yet very well how this disease works here and how is the dynamic in our environment here in the US. Okay, a little bit of the life cycle. So as I mentioned, it's an eye worm. So you're going to find the adult worm in the eyes of the host, specifically in the conjunctival sac and lacrimal duct of those hosts. And how the transmission happens. So first we need an infected host and the vector. So the vector, the fruit fly Portica variegata, are going to feed in the lacrimal secretion of an infected host and is going to ingest this first stage larvae of Thalassia calypida. Inside of the fly, this larvae is going to migrate to the reproductive system of the fly and going to molt to L2 or second, third stage larvae, and L3, third stage larvae, that is the infected form. When this infected fly feeds again in another host, they transmit the L3 to the eye of the new host. And in the eye, this L3 is going to move to L4, L5, and adult, and everything starts again. So it's a very simple cycle, but we still have a few questions about the vector. So far, the only vector that was confirmed to transmit the Thalassia was Portica species, as I mentioned, but in vitro, other fly species also showed like potential to be a vector as well. But in natural conditions, only Portica variegata is involved. Clinical signs. So this picture is a little bit, yeah, sorry. So clinical signs, most of the animals, they are asymptomatic or have subclinical infections. So they do not present any clinical signs. And when they present clinical signs, they are not specific at all. So it's very easy to overlook Thalassia infections or eyeworm infections in general. For Thalassia specifically, and I'm going to show some pictures about the worm, but Thalassia has a specific cuticle that with a specific morphology, and this morphology is what causes irritation in the eye and produce those clinical signs when present. But this, again, this is a hypothesis. We need more research on this. But which kind of clinical signs you can see in both humans and animals? Could be very mild, like conjunctivitis, itching, ocular discharge, excessive lacrimation, nothing specific. When we have a like mild or severe infection, it's when you start to see like a more concerning clinical situation that can cause, the Thalassia can cause ulcerations in dogs, in animals we didn't see in humans so far, or even secondary bacterial infection. Here I have a video that I don't know if it's gonna work, I don't think so, of the worms swimming, yeah, here. Yeah, so this is Thalassia. And what it's interesting about Thalassia and what's make like a huge difference between Thalassia and Onchocerca, it's because Thalassia, usually it's very easy to see. If you do like a eye exam, the clinician are gonna see the worm easily because they are free in the tears film most of the time. But you need to do an eye exam because as you saw here in the picture, it's not uncommon to have Thalassia worms behind the third eyelid. The difference between Thalassia and Onchocerca is because Onchocerca it's a little bit more tricky, and Onchocerca causes nodules in the ocular membrane. So it's not very easy to identify as a worm. So most of the time we detect Onchocerca doing like histopathological exams because veterinarians and humans pathologists as well, clinicians, they believe that it's a tumor or a specific or any other specific nodule, but they do not think about parasites. So during the histopathological exam, you can see the worm, and Onchocerca has a very unique cuticle, so it's one of the few parasites that we can identify in histopathological exams. But Thalassia, it's easier than Onchocerca. So usually if you do the eye exam, you are gonna see them. Oops. Oh my God. Okay. A little bit about the epidemiology so far in the US. So as I mentioned, Thalassia callipida, it's endemic in Asia and a lot of countries in Europe. But here in the US, the first case was detected in New York in 2020. More interesting than this, the vector, the Portica variegata, was detected a few years before by accident for the first time as well. So we first had the vector, and after that, we had the first case of thalassiosis in a dog. A few years after that, a number of different cases were detected in dogs and cats from different states, from New York as well and surrounding areas, including New Jersey and Pennsylvania. No, Pennsylvania, the first time was not detected. But recently, we found this second paper. Let me bring the laser again. This second paper, bring like all those cases in dogs and cats, was around like 10 cases in total, including dogs and cats, most of them from dogs. But when I saw this paper, I was thinking about like wild animals, because in Europe, we know wild animals, wild carnivores play a huge role in the transmission of the disease. And a number of different wild carnivores over there are reported to be infected or to be susceptible to thalassiosis as well. So when I saw this paper, I was like, we are not looking for wild animals. So I did what I do best, that is professional gossip. And I asked my colleagues about potential eyeworm infections in wild animals. And we found the first case of thalassiosis in a black bear was in 2023. And they told us they actually saw 3 more cases before this one, but they saved only one sample from only one animal, unfortunately. But we could confirm the Thalassia callipida infection in this bear using like morphological and molecular analysis. So it was very cool. After that, because I'm obsessed with Thalassia already, and I was very curious about like how the disease is happening here and which kind of factors are involved with thalasiosis in the US, I decided to go deep and investigate more. And I was looking to answer specifically few questions, at least initial questions. First, identify whether Thalassia callipida infections occurs in the US, if it's only restricted to those states or we are seeing in more states. Second, check for their genetic diversity because Thalassia callipida has different haplotypes. In Europe, we have only one, but in Asia, we have more than 20. And we don't know if all those haplotypes produce like different clinical disease or they are more related with some specific host population. So all those questions were in my mind. Also investigate host susceptibility. So which kind of hosts are susceptible or are more with more risk to get infected with Thalassia callipida. Identify potential environmental and host risk factors as well. And of course, provide insights into the epidemiology of thalassiosis here in the US. So for that, we decided to start with a small project. And we asked to clinicians and ophthalmologists across the US for any suspect or confirmed case of eye worm infection. I was not even specific about Thalassia or Onchocerca, and I included a picture of both just in case. We got a number of different cases from Onchocerca, not from here, from Northeast, but was very, very interesting second project. But we also got a significant number of Thalassia positive case from the Northeast. And we also asked for them to fill a form for us with a medical history of this animal and demographic info as well to try to find any association with the thalassiosis. To get those worms, we used like national advertising, basically with scientific and CE events, but the most effective one was using like the listserv. I used VetPDX, the Veterinary Parasitology Diagnostic Networking, and the Veterinary Ophthalmology listserv as well. For the parasite identification, and here's our beast, Thalassia callipida, Thalassia species in general, but Thalassia callipida specifically, it's very easy to identify only with morphological analysis. So using the morphological analysis, we basically identify the species at the parasite at species level. We also differentiate male and female worms, and we count the number of worms that were collected at that time to have any association of number of worms and clinical signs. So which kind of morphological characteristics we use to identify Thalassia, it's described here. First, the very characteristic deep cup-like buccal shape here. This is the anterior end of the worm, and this is the buccal capsula. So it's very, very unique and from this group of parasites. The second one that makes us identify them at species level, it's the location of the vulval opening that it's anterior to the esophagus intestinal junction. So this is the esophagus and this is the intestine, and here's when they meet each other, and the vulval opening usually it's located at the same location or before. Thalassia californiensis, the other species that we have here in the US, the vulval opening usually it's after this junction. And the last one is the number of transverse cuticular striation. So if you see, I don't know if you're going to be able to see here far away, but Thalassia callipida has very thin, very delicate striation across the body. And we count them because Thalassia callipida has more than 120 striations in 100 micrometers of distance. Thalassia californiensis, the distance between those striations, it's much higher. So it's very easy to differentiate them. And Female from male worms, it's the easiest part. In this case, you can see like a picture of a female worm full of larvae inside. So usually they produce eggs, but the eggs hatch inside of the female worm. So what the female worm release to the environment, to the eye secretions, it's the already well-formed L1, so first stage larvae. For molecular analysis, we also did molecular analysis for confirmation and also to get more information about the haplotype. We target the COX-1 region because it's the region that we have most information from other countries as well. But we used conventional PCR. It was not fancy at all. We also did GIS analysis. We basically, when available, it's not always available, we used the animal, the patient location for this analysis. And we collect, like, we use the data sources for each geographic layer was used from different sources, from different places. And we focus more on elevation because in Europe, some papers notice that elevation is directly related with the vector distribution and consequently with the clinical case as well. Vegetation also, and urban areas. We also check for other things, but the main goal here was these 3 points. Okay, results. We got in total so far 37 samples. From these 37 samples, 13 was considered like archival samples. So were samples from Cornell mainly, collected from 2021 and 2024. And we got only worms that were preserved in ethanol. In our lab specifically, we got 24 fresh cases. From 24 to 25, we are still receiving more cases. We're still collecting more cases, but at some point I need to stop. And in total, we got 37 samples. So far, New York, New Jersey, and Pennsylvania are the most prevalent states for thalassiosis. They had similar prevalence of the disease. Most of the cases were from dogs, 34 cases from dogs, and only 3 for cats. That was kind of expected. We got 1 case from the archival samples that were for California, but this dog was living here in New York for a very long time and recently moved to California, so we didn't count as a case from California, and was more related with a transportation than a autochthonous case. Demographic data. So most of the dogs, positive dogs, were large dogs, medium to medium large dogs. We have a very, very few cases in small and toy dogs. From the breeds, most of the breeds are from the German Shepherd group of dogs and sporting dogs, so Labrador dogs. And also after these 2 groups, mixed dogs, but all of them large dogs with a mix with pit bull. They were also positive for thalassocalip, very prevalent in this population. What it's kind of expected based on like animal behavior and also susceptibility to vector-borne disease in general, not only thalassiosis. What is most interesting, it's the majority of the dogs were senior dogs, so dogs with a 6 year or more. Very few cases were found in puppies with 1 year of age or less, and around 30% of the cases were from adult dogs, from 1 to 6 years old. This was not a surprise to me because senior dogs, so old dogs, are more struggle to avoid vectors in general. And since this fly needs a specific time to feed on the lacrimal secretion, so senior dogs tends to be less reactive two vectors or in comparison with the younger dogs. Male dogs were also more susceptible than female dogs, significantly more susceptible than female dogs. And all the three cats, I didn't include the cats here because we only got three, but all the three cats were male cats. The molecular analysis confirmed that all the species were Thalassica, and most of them was almost 100% similar with all the Thalassica sequence available at the gene bank, using only blast analysis in this case. And the phylogenetic analysis here, you can see that all cases in the US were clustered with a case from Europe. So this makes me think that the haplotype here in the US, it's the same that we have in Europe. So the haplotype 1. The GIS analysis was very interesting, although we are still working on this and we still need more analysis, more deep analysis about this. But what we have so far is that first, only 25 cases were included in the GIS analysis because all the other cases, unfortunately, the zip code or the address were not available. But all the Thalassia cases seems to be located mostly in peri-urban areas. What makes a lot of sense in my mind, especially because wildlife. And another thing that was very interesting, it's the vegetation around or near those caves that most of the time was near wooded areas with deciduous trees with like oak or hickory trees. And this is very similar to Europe as well. So they developed they did a few studies over there looking for the vector itself, and most of the vectors were found in areas with oak trees as well. So for some reason, the vector has some attraction for oak trees. Regarding the elevation was the other factor that I was looking for, again, based on data that we have from Europe. Most of our cases were at low altitudes between 150 to 350 meters above sea level. What is very, very different than Europe, because all the cases that we have there, the majority of the cases that we have there were like above 800 meters above sea level. What it makes me think that first, maybe altitude is not a very good parameter for thalassiosis, or we have a different environment scenario here, and maybe there is some animals or birds that feed the vector in higher altitudes that are not in Europe. And maybe at lower altitudes that we are lacking this natural vector control, it's when we are see a high number of the vector, and as consequence, like high numbers of the clinical cases as well. So after saying like all this information, although this is a very initial project and we have a low number of cases so far, we can start to connecting all those dots and creating hypotheses for the next steps. The first one is the genetic diversity, of course. As I mentioned, Thalassia callipeda haplotype 1, that it's the same from Europe, was the only one detected here so far. It's presumed that Thalassia callipeda is not native to the US. We are not 100% sure about this in Europe and Asia. But in the US, we are almost 100% sure because of this, because the haplotype is identical to Europe. So reinforced the idea that this disease was imported a few years ago. And second, because of the timeline of the infection, of the distribution of the disease. Because first we detect the vector for the first time. Portugal variegatus was detected in New York in just fly traps by accident. And in 2016, and a few years after, when the vector was well established in the US, we got the first case of Thalassia callipida in a dog. And since then, we are seeing more and more cases in dogs and cats, which is very concerning because at some point we are going to start to see in humans. The urban cycle. So among all Thalassia species, Thalassia callipida has the widest range of definitive hosts, for sure. Thalassia callipida, as I mentioned before, we have a report of a number of different carnivores in Europe. Here in the US, we know so far that bears, black bears, dogs and cats are susceptible to the disease, this disease, but I believe more carnivores, special small canids like foxes, they are also probably play a significant role in the transmission of the thalassiosis. As we also saw in our project, Thalassia callipida is commonly found in dogs. This is also very similar in Europe and Asia. And there is like a number of reasons why dogs are more susceptible. First, increased mobility, hunting behavior, mass movement of animals, people. So everyone is traveling, parasites as well. And dogs become more susceptible to those situations. We saw lower prevalence in both small-sized breeds and younger dogs, which epidemiologically makes a lot of sense. All case seems to be concentrated in peri-urban areas, and vegetation seems to play a role on the vector distribution. But there is still have a number of different questions that I would like to answer, like monthly preventives, are they effective or not in preventing thalassiosis? We do not have this answer. Some of our dogs, our positive dogs, were on preventives, heartworm preventives, and also ectoparasiticides preventives. So this seems to not be effective against flies, but we cannot rule out vector resistance as well. So antihelminthic and also arthropod resistance to all the antiparasiticides that we have available, it's an issue in the US, not only with the Thalassia, but other worms, hookworms, roundworms, tapeworms. So we need to investigate this more. And another question is about this sylvatic cycle. So far, we only detected thalassiosis in black bears. So we need to investigate and we need to keep looking in other carnivores to understand the dynamic of this parasite, specifically on those environments, and also to understand how these parasites affect those populations, because preservation is also another topic that we should consider. Prevalence and public health considerations. So Thalassia callipida should be considered endemic in the Northeast US for sure. I don't think this parasite is exotic anymore. So this should be in the differential list of veterinarians and also physicians, especially ophthalmologists, because again, at some point we are going to start to see in humans. We need to find a better way or an efficient way to have to prevent this disease in animals specifically. So prophylactic measures is still something that we are looking for. In Europe, there is some research about the potential use of regular preventives, GI parasites or heartworm preventives as preventives for thalassiosis as well. But all those papers are based on clinical cases in endemic areas. So it's very tricky to interpret. So I don't think we have a very good research data to confirm that one specific drug or compound is effective in preventing telesiosis so far. The infection rate in animal is probably not yet high enough to lead to many human cases like we are seeing in Asia and Europe, but based on like how fast this is increasing in the animal population, probably we are going to see this in the next years in humans as well. And another thing to keep in mind is that human telesiosis is often overlooked or misdiagnosed, especially in vulnerable populations. We know that vector-borne disease in general are more prevalent in low-income or middle-income countries. So I believe low-income communities here in the US are going to be more susceptible to telesiosis, especially because lack of access to healthcare and also because of environmental and economical factors. And this brings the One Health concept as well, associated with vector-borne disease. So for those that doesn't know what One Health means, One Health is just a concept of interdisciplinary action between different fields, not only veterinaries, not only physicians, so not only veterinary medicine or human medicine, but also economists, sociology, and epidemiologists, biologists, to integrate actions, especially with those in the government, to integrate actions to prevent disease, especially zoonotic disease, from that could affect humans and also animals. Besides that, so vector-borne parasitic zoonoses have an impact on animal and human health. We all know this. It's well established. But it's also important to highlight that vector-borne disease in general also represents a major threat to any country's economy. And we have evidence of that because most of the vector-borne disease, as I mentioned before, are present. The high incidence of vector-borne disease are present in low-income areas, low-income countries or middle-income countries. So the next step, I believe, to include telesiosis, to integrate telesiosis or any vector-borne disease, to be honest, to the One Health concept, it's education and disease awareness, especially between vets, among vets, physicians, and also animal owners. Everyone play an important role preventing the disease and the spreading the disease. Early diagnosis and efficient treatment prevent complications. In telesiosis, we can avoid more severe clinical signs and also unnecessary use of antibiotics. Disease surveillance, it's another step that it's important to have, and vector control programs as well. So we need to develop surveillance policies for traveling animals, especially. We had this case from this animal traveling from New York to California, infected with thalassia, and was only diagnosed over there in California. So this is another thing to keep in mind. And everything and all those policies should focus on restricting the distribution of the parasite, not only thalassia, but any other zoonotic parasite across states, but also to other countries. As future directions, I would like to do a large-scale prevalence study. So I would like to continue collecting samples and maybe increase our number to have a more robust analysis, and maybe we could extract more information, especially epidemiological information. We still need to understand the role of wild animals in this transmission and distribution of telesiosis. We also need to know the real host range of telesiosis here in the US. I don't think it's only black bears are susceptible to thalasiosis or are involved in Thalasia callipida infections in the wild environment. We need also know and understand the impact of thalasiosis on this population, different population. Domestic animals will have a better idea, and I think it's easier to have more information about it, but it's very hard to work with wildlife. And the impact that a thalasiosis could cause to those animals, it's hard to tell and it's hard to measure. Vector distribution, also with prevalence, of course, is a vector-borne disease. So if you do not have a vector, we do not have a disease. So we need to work with both together, detection of thalasia and also the vector. And of course, exposure and risk of transmission for all those different populations, humans, domestic animals, and wild animals. This work was a collaboration with a lot of people. Of course, I'd like to thank my lab, Clinical Infectious Disease Lab. They are all amazing. I could not do it, and I could not find time for research without them. But especially Jackie, that's here today, that did all the molecular analysis and the collection step as well. Isel, it's a vet student at Penn Vet that did the GIS analysis. Cornell University with money. Tiago, my husband, he's an urban planner and the only person capable to work with the GIS. I have no idea how to use the GIS. So he's helping me a lot to navigate in this new software. And of course, all the veterinaries and clinicians that contribute and donate the sample and also share information about their clients. And I have to take any questions. Yes.
So that was super interesting talk. Are the vectors primarily outdoors? And so like, are the dogs that you're in your study that were getting infected, are they spending a lot of time outdoors?
Yes. Yes. We, so the question is if the vector is more prevalent in outdoor areas. Yes, Portuga variegata, it's a fruit fly that do not go inside the house usually. So all those animals have access to outdoor areas. Yes.
I have a question about sort of like, you were talking about like the spread of it, right?
Mhm.
And so, so sure, there's like the aspect of maybe just the dogs are outdoors more and like they are exposed to these environments more versus the cats maybe more inside.
Mhm.
But is there like a component of like the actual, you said it like kind of feeds on the tears and the stuff
Mhm.
in the tears, are like differences between like animals in their tear components that could be like leading to this?
It's totally possible, but we do not have research for that. Again, all the information that we have, it's from Europe or Asia, and everything is based on clinical case. So for sure, we need like maybe a laboratory scenario to control all the other factors to have like this specific information. But it's a great question. Yeah. Yes.
Awesome, super cool talk.
Thank you.
Were the cats outdoor cats?
Yes, all 3 cats had access to outdoor areas. I forgot to mention that. Thank you.
And then my second question is this the same worm that is like on the US-Mexico border that I've been hearing about and people are worried about?
No, this is a screwworm
Okay.
that causes myiasis. It's Cochleomyominovarex. It's another fly. Yeah.
Different fly.
Different fly. That fly eats meat, flesh meat. So it's a different
So-
one.
More scared about that one, worried about.
Yeah.
That's
Yeah. Different vectors. Yes.
Cool talk, deeply horrifying. Questions, what are the larvae eating in the eyes? Like, are they subsisting on tears or are they eating actual like tissue?
Great question. I do not have this answer. Based on like information that we have from other filarial nematodes, I believe they are feeding on bacteria, and also they have their own source of food. When the female worm release the larvae, all the larvae has like a ex how can I say that? Have like a saved amount of energy, food energy inside of the intestine. So could be a combination of both factors, if that makes sense.
And second question, you said, I think that this one is particularly irritating to the eye. Like, is there something about like chemical that's irritating?
Yeah, it's the striation. So the morphology of the striation, it's on a hypothesis. They could not confirm this so far, but it's on a hypothesis that the striation of the worms cause this irritation to the eyes. However, there is a number of different papers showing that the number of worms found in the eyes, including our study, are not necessarily related with severity of clinical signs. So we had dogs with more severe clinical signs that only have like few worms. At the same time that we have dogs with a large number of worms that was present like mild clinical signs or even asymptomatic. So it's not clear yet. Yes.
What are the clinical signs in humans similar to?
Yes, they are all similar, but in humans usually do not get that bad. Because humans, it's better in talking about we have something in my eyes that it's not comfortable. But one thing that I'm thinking, and I believe it's going to happen, it's that kids and elderly people are going to be more susceptible to vector-borne disease for the same reason. So we can mimic at somehow the findings here with the dog population and human population. But in general, all the reports that we have in humans from other countries, it's not that bad with the clinical signs. Usually it's more mild and or even asymptomatic case. Yes.
Okay, so for hosts, we talked a lot about carnivores, but you also had a picture of a cow. Is there a preference for what type of host?
It's related with the Thalassia species. So Thalassia gutturosa, it's one that infected ruminants, not Thalassia calypida. Thalassia calypida, it's only in carnivores and lagomorphs.
Yeah, so you mentioned that this worm is like the vector is the male fruit fly. So why, like, is there any like evolution advantage to that? Because I mean, for other vectors like mosquitoes, the female kills everything and everything. So, right, what do you think is why?
Great question. $1 million question. I don't know. What we know, it's like female flies are also susceptible to Thalassia. So they can feed, at least in the laboratory environment, they could feed in lacrimal secretions of humans and dogs. And they also got infected with thalassiosis. But in natural conditions, only the male fly feeds on mammals' lacrimal secretions, but we don't know why. The hypothesis is because of the level of protein that the male requires for reproductions. It's much higher than the female fly. So this fly do not feed like all the time in lacrimal secretions. It's only during specific time they believed is related with reproduction. This is why.
So it's, well, not just a follow-up question. I just wanted to clarify. So it will be like the more common encounters because the male fly would become like the animals versus the females that also have that bugs.
Yeah, good point. Yeah. Yes.
Is this naturally resolving in dogs?
Mm-mm.
So there you couldn't really figure out there's like a seasonal aspect to it because they could last there for over a winter.
So it's related with the vector, right? So we don't know here. Everything that I'm gonna say, it's again related with Europe specifically. We know in Europe, the vector Portugal variegata has a peak during the summer, during July. So the whole life cycle takes around a month or a little bit more. So all the clinical signs, or at least infected dogs, we start to see after the summer. So the peak of case is during the fall and beginning of winter. And this was what we saw in this past year. During the summer was very quiet. Spring and summer was very quiet. We didn't receive any case, but during the fall and winter was a significantly high number of cases, at least one case every week in some months.
So it lasts in the animal for the flies to pick it up again the next year?
Say, oh, how long it takes to produce larvae? It's what you're saying? Around a month. Yeah, in the eye, around a month. Did I answer your question?
Yeah.
Okay. Any other question? Yes.
Is there anything known about endosymbionts of either the worms?
No. Unfortunately, I'm the only person interested on this. But I'm gonna try to talk with maybe USDA because they have this surveillance program for fruit flies in general, but they are looking for other species of fruit fly that have like economic impact in farms. So this fruit fly, it's not important, economically not important, but maybe we can work together and coordinate, because if they collect even by accident, any Portuguese species, we can check in the lab and have a better idea where is the disease is located. Okay, thank you.
So I was thinking that like over the winter, so they pick it up, hold it, fly pick it up in the spring. and
Worst
then
case
pick it
scenario.
up. Like, kind of like a nugget.
So they like.
Try to-
float it in.
Yeah.